Now we are going to discuss about the structures, different structures of cells, namely the prokaryotic cell and the eukaryotic cell. So the most important things that are needed to be discussed in this area is that the students need to know the difference in their structures. Okay, so it is very important that students know the differences in their structures, because this is actually one of the most common questions that is being asked in IB, especially in the essay question. So by actually showing the students how the structures are being drawn, or making a diagram of how the prokaryotic cell and eukaryotic cell is being drawn, then we might be able to show the differences to the student. Apart from that, apart from showing the structure, of course, we need to label them. Okay? So the labeling the structures is also a part of one of the questions, or one of the key areas that is being asked to IB students in. Apart from that, once that the students have been able to label the structures, then they should also know the function of each structure. Okay, so of course, for example, there would be a difference in the cell wall in prokaryotic cells and cell wall in plant cells. Okay, first of all, in plant cells, cell walls are being used to provide a rigid structure, but for prokaryotic cells are being used, the cell walls are being used to for protection itself. Okay. Another important key area that is needed to be learned by students is that they need to know the comparison, or the differences between prokaryotic cells and eukaryotic cells. So we can show the comparison between the two structures by the most effective way that we can show this, if we are able to show them into a table, okay, or into a matrix. So for example, by writing down the basic features that is present only in prokaryotic cell and eukaryotic cell, then we might be able to give the comparison. So showing the main features. Okay, so under eukaryotic cell, okay, we can now elaborate that eukaryotic cell has two types. We have plant cell and animal cell. So under animal cell and plant cells, there are still differences. So we can also show the comparison between the two if we are able to write them down into a matrix. Okay. Structures. Okay, so by using a matrix, you can also elaborate on the features that is present for both animals and plants. So for example, the presence of cell wall. Cell wall is only present obviously in plant cells, whereas for animal cells they are absent. The presence of chloroplast. Chloroplast is an organelle that can only be found in plant cells, whereas it is absent in animal cells, such as that. Another thing, yeah. So basically, by writing down the differences between the animal cell and plant cells and writing down the main features of these structures, we might be able to discuss wholly on the prokaryotic cell and eukaryotic cell. The next area that we need to discuss with students is the subtopic about membranes. Actually, membranes is actually a part of the structures that is being discussed in the previous topics in prokaryotic cells and eukaryotic cells. But we can also elaborate to the students, we can tell the students that we can further discuss this area, okay, in this separate topic. So here in membranes, okay, Okay, the most important thing that we need to discuss here is to relate the structure with the function. So by relating the structure to its function, then the students would be able to understand how the movement of substances is occurring in membranes. So here in membranes, we need to discuss the structures or the components that are present in membranes. Okay, for example, in the case of, we have the phospholipid bilayer. We have to discuss why is it arranged into a bilayer. Okay, we should also discuss as well the chemical structure of a phospholipid. So phospholipid is actually made up of a combination of a glycerol group. We have the fatty acid chain, and one of the features that is present in a phospholipid is the presence of the phosphate group attached to the glycerol group. So by having the phosphate group in the glycerol, then it allows, or it gives the property of the phospholipid bilayers in becoming in being hydrophilic. Okay, so we need to write down their functions as well. Okay, so in the case of another structure, we have, let's just say an example, structures present in phospholipid. We have the membrane proteins. Okay, membrane proteins, since there are different types of this, we need to elaborate the function of each. For example, we have the membrane protein called channel proteins. Okay, channel proteins, the function of this is that it allows diffusion, easy diffusion of substances from a higher concentration to a lower concentration. But in the case of a pump protein, since it's called pump, then it needs energy to move substances from a lower concentration to a higher concentration. So we need to relate the structure with the function as well. So another thing that we need to take note of is the properties, okay, of phospholipids. Okay, as I've mentioned a while ago, that by discussing the chemical structure of phospholipids, we will be able to pinpoint the reason why the structures are, the phospholipid are hydrophilic and hydrophobic. Okay? So by having this properties, hydrophilic and hydrophobic properties, then we can come to a conclusion that the cell membrane or the plasma membrane as a whole is a semi-permeable membrane that only allows certain substances to enter the cell or exit the cell. Okay? So the next area that we need to discuss about membranes are the movement of substances. Okay, substances such as solutes, okay, or nutrients, substances through membranes. So we will discuss the things about diffusion, osmosis, active transport, facility to diffusion, and of course we also need to differentiate the differences between diffusion, simple diffusion, and facilitated diffusion. Okay, so we also need to discuss about bulk transport. So in this case, this entails the transport of substances that are in greater amount. For example, if the cell is producing a specific protein, then we need the cell needs to use vesicles or sacs that that is being produced by the membrane-bound organelles such as the Golgi apparatus and the RAF-ER, and then they are being moved out through exocytosis. So the next part is, of course, the transport of materials. So through the transport of materials, actually we can also relate this transport of materials in the previous area or the previous point that we have discussed, use of vesicles. So since we have already lined out all the things that is needed to be remembered by students in this area of cells, then we might be able to encompass the things that is needed to be discussed.